This presentation on sequencing by synthesis was created for the University of Alberta course Medical Laboratory Science 480 and is presented by Dr. Karen Matika. This presentation was created using resources created by Andrew Workman and Illumina. Copyrighted material contained herein is reproduced under subsection 29 through 29.4 of the Canadian Copyright Act. This presentation is available for your individual use. Further distribution may infringe copyright. By the end of this presentation, the learner should be able to describe library preparation, describe and interpret a bioanalyzer trace, calculate the amount of library required for instrument loading, describe a flow cell and a cluster, describe traditional cluster generation, describe pair-end sequencing, describe Illumina sequencing by synthesis, interpret 2- and 4-channel sequencing data, and describe a Q-score. As you may have noticed from the learning objectives, sequencing by synthesis consists of many steps. The figure on this slide outlines the steps of next-generation sequencing using the Illumina platform. We start with sample preparation. The nucleic acids need to be extracted from the sample and quantitated. A library is then created from the purified nucleic acids by ligating adapters onto the ends of the nucleic acids and by creating clusters on a flow cell. Following the creation of the library, the sequence of the nucleotide is determined by fluorescence signals. And the final step is data analysis. Preparing a specimen for next-generation sequencing is laborious and expensive. Next-generation sequencing requires a large amount of pure DNA. Illumina library preparation requires 1 microgram of DNA. For superior accuracy, DNA is quantitated using fluorescent dyes. Nucleic acids isolated using commercial kits are usually of a good quality, but the saying junk in equals junk out really applies here. Illumina recommends using an agarose gel to determine whether or not there is contamination of your DNA before proceeding with library preparation. Impurities such as detergents or proteins are visible as smears on your DNA gel, and RNA is often visible at the bottom of a gel. A ladder or smearing below a band of interest may indicate nicking or other damage to the DNA. Once you are sure you have sufficient good quality DNA, you can start the library preparation. The figure on this slide outlines the steps of library preparation. The first step is mechanical fragmentation. The genomic DNA is sheared into fragments of approximately 300 base pairs by ultrasonication in an instrument called the Covaris. The shearing of the DNA may result in 5' or 3' overhangs, which brings us to step 2, end repair and phosphorylation. To facilitate the addition of adapters, the ends of the fragments are repaired using a DNA polymerase to fill in the 5' and 3' overhangs. The 5' ends of the fragments are also phosphorylated using the calf intestine phosphorylase. Following that is step 3, adenylation. The 3' ends of the fragments are then adenylated to create a 3' poly A tail. The poly A tail facilitates the ligation of Illumina adapters to the DNA fragments, as the adapters have poly T tails. It's important that you remember that while the figure shows a single double-stranded DNA fragment, multiple fragments covering the entire genome are being ligated to adapters. Once the adapters have been ligated to the DNA fragments, it is possible to amplify the library to increase the amount of material for sequencing. Following adenylation of the 3' ends of the DNA fragments, Illumina adapters are ligated via their poly T tails. The ligated adapters have multiple sections as shown here on this slide. On the right-hand side of the adapter, the nucleotide sequence is labeled P5, index RD1SP. The P5 sequence is complementary to oligos that are bound to the surface of a flow cell. The index sequence is a unique sequence of nucleotides used to identify the source of the DNA fragments, for example, patient A. This allows multiple samples to be processed on one flow cell. For example, patient A would have one index sequence and patient B would have a second index sequence. The RD1SP sequence is complementary to one of the sequencing primers. Then we have the DNA insert, which is our fragmented DNA, which is approximately 300 base pairs in size. On the left-hand side, the adapter nucleotide sequence is labeled RD2SP, index, and P7. The RD2SP sequence is complementary to the sequence primer. The index sequence is a unique sequence, again, used to identify the source of the DNA fragments. Depending on the type of library prepared, this index sequence may or may not be identical to the one on the right-hand side of the DNA insert. Again, the index sequence allows multiple samples to be processed on one flow cell. Following the index sequence is the P7 sequence, which is again complementary to another set of oligos that are bound to the surface of the flow cell. After the genomic library has been prepared, it needs to be quantitated and the quality of the library needs to be assessed. The amount of DNA is measured using fluorescent dyes. Illumina recommends quantification of DNA using the qubit instrument, which uses its own fluorescent dye that is specific for double-stranded DNA. The quality of the library is analyzed using a bioanalyzer or a fragment analyzer. The fragment analyzer, or bioanalyzer, is used to calculate the size and concentration of the nucleic acid fragments from the library preparation. It is a QC step prior to sequencing. A sample of the library preparation along with 2 markers are applied to a well in a microchip. An Agilent bioanalyzer lab chip is shown on the left-hand side of the slide. Each chip contains an interconnected set of gel-filled channels that allow for molecular sieving of nucleic acids. A series of electrodes within the microchip control sample movement. Inside the chip, the library DNA and the 2 markers are separated by size. On the right-hand side of the slide is an example of a bioanalyzer trace. The upper marker is indicated by a green arrow and it is larger than the library DNA. The lower marker is indicated by a purple-pink arrow and is smaller than the library. The markers are used to extrapolate the size and concentration of the DNA library. Ideally, the library will appear as a single uniform peak. To extrapolate the size of the library, the 2 markers run with the sample are compared to a series of standards. An example of the DNA ladder or standard is shown in the top image on this slide. The lower image is an example of a poor quality library preparation. Notice there are multiple fragments between the upper and lower marker. Remember, ideally the library is a uniform size. The concentration readings from the fluorescent measurement of the DNA and the bioanalyzer data are used to determine the library concentration in nanomoles per liter. The concentration in nanograms per microliter is multiplied by 10 to the power of 6 to determine nanograms per liter. This value is then divided by the quotient of 660 over the size of the library. The concentration of the library is then adjusted to 4 nanomoles per liter, and a total volume of 5 microliters is prepared for sequencing. After the library has been prepared and assessed for quality and quantity, the next step is cluster generation. The library is alkaline denatured into single strands, neutralized, and applied to a flow cell. The flow cell is a glass slide with multiple lanes. there is an image of a flow cell on this slide. The lanes are coded with short DNA sequences that are complementary to the P5 and P7 section of the adapters that were ligated to the library. When the library is flooded across the flow cell, the library will hybridize to the lawn. Illumina does produce another type of flow cell that has wells, but for simplicity, I will only refer to this type of flow cell. The single-stranded library hybridizes to the flow cell via the P5 and P7 sequences on the adapter. The individual strands of hybridized DNA are amplified into clusters by a process known as bridge amplification. Each cluster represents thousands of copies of the same DNA strand in a 1 to 2 micrometer spot. After the library has hybridized to the lawn on the flow cell, a complementary strand of DNA is synthesized using a DNA polymerase. Now, the next thing that they do is not very intuitive. They denature the double-stranded DNA and wash away the library strand that was hybridized to the lawn. This leaves only a new strand of synthesized DNA bound to the flow cell at its 5' end. The single-stranded DNA bound to the flow cell is the starting point for bridged amplification. The image on the right shows single-stranded DNA standing up. Really, it's not. The free 3' end of a single-stranded DNA molecule is more like a piece of cooked spaghetti falling over, and when it falls over, it's going to encounter another single-stranded DNA molecule that is attached to the flow cell. This is complementary to the other adapter, and it hybridizes. So in this image, initially, the DNA strand is captured by hybridizing to the P5 sequence on the flow cell and the P5 adapter sequence, and then it falls over, and the other end of the molecule, the P7 adapter, hybridizes to a complementary oligo on the flow cell. The complementary strand is then synthesized using a DNA polymerase. Both strands of the molecule are attached to the flow cell. The 4 images on this slide depict bridge amplification. The image in the top left corner of the slide shows a double-stranded DNA molecule that was synthesized by a DNA polymerase. This double-stranded DNA molecule, or bridge, is denatured, resulting in one strand hybridized to the flow cell via the P5 adapter, and the other strand hybridized to the flow cell via the P7 adapter. This is shown in the image on the top right of the slide. Next, the image in the lower left depicts the free ends of the single-stranded molecules hybridized to the oligos on the flow cells as they are extended by DNA polymerase. And the image in the bottom right shows the resulting double-stranded molecules or bridges. These double-stranded molecules of DNA or bridges are denatured into single strands, and the process is repeated. This is bridge amplification. Following bridge amplification, the double-stranded DNA is denatured. This will result in single-stranded DNA being hybridized to the flow cell. This is depicted in the image on the left-hand side of the slide. In order to sequence, we need to remove one strand so that we're not getting cross signals. So the next step is to cleave off the reverse strands and wash the flow cell to remove those strands. The removal of the reverse strands is the last step of cluster generation. The next step is sequencing. This form of sequencing is called sequencing by synthesis, and it has some similarities with Sanger sequencing. The sequencing primer anneals to the read1 primer binding site, and labeled dideoxynucleotides flood the flow cell. Remember that dideoxynucleotides lack a 3' hydroxyl, so only one nucleotide can be attached at a time. Photographs are taken by a camera attached to a microscope. These pictures capture the signal given off by fluorophores attached to the dideoxynucleotides. Following image capture, the dyes are cleaved off the nucleotides and the dideoxynucleotides are enzymatically converted back to deoxynucleotides, which have 3' hydroxyl groups. Then the next round of sequencing begins, and the flow cell is again flooded with dideoxynucleotides. There are different chemistries for doing the labeling of the nucleotides. We have a 4-di chemistry and a 2-di chemistry that we'll talk about. This slide gives a closer look at the process. Starting from the left-hand side of the slide, the primer attaches, and the DNA polymerase adds a complementary labeled dideoxynucleotide. In the 4 chemistry sequencing, the camera takes 4 photographs to capture the signal off of the fluorophore, which was attached to the dideoxynucleotide. Each dideoxynucleotide is labeled with a different colored fluorophore. Following image capture, the fluorophore is cleaved off the dideoxynucleotide, and the dideoxynucleotide is converted to a deoxynucleotide. Thus, we can then repeat the process and add a dideoxynucleotide, take a picture, cleave off the fluorophore, convert the dideoxynucleotide into a deoxynucleotide, and repeat. In 4-channel sequencing, each dideoxynucleotide is labeled with a different colored fluorophore. Subsequently, 4 images are captured each time a nucleotide is added. Color will be observed in only 1 of the 4 images. An alternative to the 4-channel chemistry is the 2-channel or 2-di chemistry. The process is very similar. The primer attaches to the DNA, and the DNA polymerase adds complementary labeled dideoxynucleotides. This time the camera will take only 2 photographs to capture the signal of the fluorophores attached to the dideoxynucleotides. The fluorophores are then cleaved off of the dideoxynucleotide, and the dideoxynucleotides are converted to deoxynucleotides. Then the process repeats. In the 4-channel or 4-di chemistry, each dideoxynucleotide was labeled with a different colored fluorophore. In the 2-channel or 2-di chemistry, 2 images are taken. Thymines appear green. They are labeled with a green fluorophore, so therefore in the green capture camera they are visible. In the second camera, which is red, they are not visible. Cytosines are labeled in red. They are not observed with the green camera, but they are seen with the red camera. Adenines are labeled yellow. They are visible in the green camera and the red camera. Guanosines are not labeled with a fluorophore, and therefore they appear dark in the green camera and with the red camera. Therefore, we are able to differentiate the 4 dideoxynucleotides based on their labeling system. This slide is a summary of the differences between the 4-channel and 2-channel chemistries. Again, in the 4-channel chemistry, each dideoxynucleotide is labeled with a different fluorophore. In the 2-channel chemistry, thymine, cytosine, and adenine are labeled with fluorophores, and guanosine is not labeled. For most applications in the clinical lab, we want to sequence the forward strand and the reverse strand and match them together to ensure that we have an accurate accounting of the DNA sequence. This is called paired-end sequencing. In paired-end sequencing, after we have the sequence read with the read 1 primer, we need to generate the complementary strand and sequence it. In order to generate the complementary strand of DNA, the strand of DNA created during sequencing must be removed. This is depicted in the image on the left-hand side of the slide. Next, the 3' end of the attached single strand of DNA hybridizes to the flow cell via the adapter at the 3' end. The 3' end of the flow cell oligo that it is attached to serves as a primer for DNA synthesis of the complementary strand. This is shown on the right-hand side of the slide. The extension of the DNA from the oligo on the flow cell lawn results in a double-stranded DNA molecule. This is depicted on the left-hand side of the slide. While the image shows only a single molecule of DNA, it's important to remember that at this point you have already created a cluster. So this extension step will result in a cluster of reverse strands. Next, the double-stranded DNA is denatured, so only the single-stranded DNA is hybridized to the flow cell. Subsequently, the forward strand is cleaved off and washed off the flow cell, leaving only the reverse strand hybridized to the flow cell. This is shown on the right-hand side of the slide. The reverse strand is now ready for sequencing. The read 2 primer hybridizes to the complementary sequence in the adapter on the reverse strand, and sequencing by synthesis begins. As you might recall from earlier in the presentation, the adapters have index sequences that are used to identify the source of the DNA. This allows us to mix multiple samples from different patients together onto one flow cell. These indexes are read during the sequencing process. So the process is you do the read 1 sequence read, then you read the index, you do pair end turnaround where you create the complementary strand, then you read the index, and following that you sequence the reverse strand. A large amount of data is created during these sequencing runs. During the run, images are captured after the addition of each nucleotide. These images are processed during the run to make base calls. The intensity of the color coming from each cluster, as well as other parameters, are used to create a quality score for each base. After the sequencing run is complete, the data is demultiplexed. That is, the sequences from individual libraries are separated via their index sequences. The DNA sequences are converted to FASTQ files and can be aligned to reference sequences. Variations from the reference sequence can be interpreted. During data analysis, a quality score is assigned to each base call. Shred quality scores, also known as Q-scores, are the most common metric used to assess the accuracy of a sequencing platform. The Q-score indicates the probability that a given base is called incorrectly by a sequencer. For example, a Q-score of 30 is equivalent to the probability of an incorrect base call 1 in 1,000 times. This means that the base call accuracy is 99.9%. A lower base call accuracy of 99% is equivalent to a Q-score of 20. This means that for every 100 base pairs that have been read, they will contain at least one error. When sequencing quality scores reach Q30, virtually all the reads will be perfect. During data analysis, the DNA sequence is converted to a FASTQ file. FASTQ files consist of 4 lines. The first line is an identifier. Typically, the identifier includes the instrument and the flow cell, as well as the location of the sample and possibly the sample accession number. The second line is the sequence. So you can see here that the sequence is TCG. The next line is a location line, so we can see we have a plus underneath the T. This indicates that the quality score in the fourth line is for the T nucleotide. When this plus sign moves to be underneath the C, we then are going to see the quality score for the call for the cytosine. This slide represents the multiplexing and demultiplexing of DNA libraries. We can see here that we have 3 different libraries, sample 1, sample 2, sample 3, and you can see by their adapters that they indicate that they are separate. So sample 1 is red-blue, sample 2 is blue-blue, and sample 3 is yellow-blue. Having unique identifiers from the index sequence allows you to pool these samples or libraries together. They are sequenced and the FASTQ files are created. Following that, the computer will demultiplex these and using the adapter sequences to put them into bins or silos of information. Following the demultiplexing step, the data is filtered to get rid of files with poor Q-scores. Remember, you are sequencing hundreds of pieces of DNA at the same time. The sequence is then compared to a reference sequence such as the human chromosome available on the NCBI website. An alignment is made and sequence variants can be identified. The next step in the process is to determine whether these variants are clinically significant or not. Dr. Hume will show you how this is done in another lecture. One of the many advantages of next-generation sequencing is that you can increase the depth of your DNA sequencing. This means you have more reads covering the same area of DNA. While this costs more money, it does increase the sensitivity and your ability to detect mutations in a population of cells. It also increases the confidence in which you can detect these mutations. In summary, Illumina sequencing, or sequencing by synthesis, can be broken down into 4 processes: library preparation, cluster amplification, sequencing, and data analysis. In library preparation, the genomic DNA is fragmented and adapters are ligated. In cluster generation, the library is hybridized to flow cells and is amplified to generate clusters. Sequencing is performed using labeled dideoxynucleotides. Data analysis is a multi-step process. FASTQ files are created, aligned to a reference sequence, and variants must be interpreted. The following references were used in the creation of this presentation.